Hello everyone, and welcome again to the third webinar in the series of 12 on berry crop nutrient and soil management. We're very excited about the program. We still have people registering for the first time. We're up to 72 people now. If you have any colleagues or associates who might be interested in participating, they're still welcome to join us. I just want to do a quick reminder. I want to point out to you in the files box on the bottom right that the webinar schedule is there that you can download. The demographics questionnaire, that's the registration form. If you haven't filled that out and emailed it back to me, please do that. And then the educator pretest for any of you who are new, there's a pretest there. Please fill that out and also email it back to me at mcm4@cornell.edu. If you would like to participate in the webinar, you are able to type comments or questions into the chat box at the bottom. That's the same place where you typed in your name and your location. Other ways to participate are up on the top of the screen. You'll see a person with their hand raised. There's several things that you can do. You can agree or disagree, raise your hand to indicate you have a question, ask someone to speak louder or softer, to speed up, to slow down, to laugh, to applaud, and all of those little icons will show up next to your name in the attendees box. On the bottom left-hand side of the screen, you'll see some web links. Northeast SARE has its own website, and it has a link there. Cornell Fruit Resources Berries is also there, and then at the bottom are the webinar archives. If you've missed any of the webinars in the series, they are recorded and you can listen to any you've missed by going to that link. Okay, we're ready to start. Our speaker for this week is Dr. Marvin Pritts, Professor and Chair of the Department of Horticulture at Cornell University, and our topic is soil nutrient management. He's going to discuss correction of soil problems, application technology, conventional nutrient sources, organic nutrient sources. Oh, I'm sorry, that's next week's topic. Liming and pH reduction. So now you know what we'll be studying next week. Marvin was born in southwestern Pennsylvania and got his BS in biology from Bucknell and his MS in biology from the University of South Carolina. He got his PhD in horticulture at Michigan State University, working with wild species of blueberries. He came to Cornell in 1984 as the berry crop specialist, with an appointment in extension, research, and teaching. Marvin works primarily with production and pest management systems in strawberries and raspberries, including food safety issues, and has consulted with berry farmers throughout the world. He became chairman of the Department of Horticulture in July 2002. He teaches career explorations in the plant sciences, hands-on horticulture, berry crop production and management, and leadership development for life scientists. Marvin?
Thank you, Kathy, and welcome all of you to the webinar. It's a beautiful day here in New York. I hope it's nice where you are as well. What we want to do today is talk about how to interpret soil tests. And the soil tests, as we mentioned before in previous webinars, are used before planting. That's where we're primarily using our soil tests. And the goal of soil testing, just to refresh, is to estimate the plant available nutrient levels in the soil. Soil tests do not tell you how much of a particular nutrient is in the soil. They estimate how much of a particular nutrient in the soil is available to the plant. So the extractant sort of mimics what a plant root might have available to them. And since you can't mimic that perfectly, people have come up with these different extractants to try to imitate what a plant root might be able to extract from the soil. Once those values are obtained, then a recommendation is made to bring those levels up to some optimum, because we don't want soil nutrients to be limiting. Typically, plants respond to soil nutrients in a curve that sort of looks like you get a rapid response at low levels, but as you get up further in the curve, the response to more and more nutrients begins to level out to where you don't get a response at all. And the goal of soil nutrient testing is to somewhat build up the soil nutrient bank so that there's never a risk of nutrients being limited. But there are some philosophies of soil testing where you want to just apply enough to where the economic gain from an additional amendment is equal to the cost of the added amendment. So let me just maybe try to show you that graphically. In some philosophies, you want to try to be right here on the curve where you're just starting to see a decrease in the response. Other philosophies say, well, we want to be up here because we don't ever want to be in a situation where we want to be in a deficient condition. And different soils labs sort of give you recommendations based on that philosophy. And probably not surprisingly, soils labs that also sell fertilizers tend to be up here at the top end, and university labs and all tend to want to be down here. It's a little bit harder with perennial crops to be in this range here where the green arrows point out, because we don't often have a chance to go back in annually and add a little bit of nutrient to bring us up to where we need. So if you look at recommendations for annual crops, they'll often be down in this range. With berry crops and perennial crops, we tend to have to find a happy medium between here and up here, because we can't go in annually and replenish very well. So the ideal is somewhere between, we just can't go in and incorporate the amount of nutrients we need after a year is up. So, except for sandy soils, the eventual nutrient supply, which is how much is in the soil, plus how much we add as fertilizer, should be sufficient for the life expectancy of the planting. And this would be looking at, maybe 5 years for strawberries, maybe 10 to 15 for raspberries, without having to come back in and annually apply phosphorus, potassium, and magnesium, calcium, and so forth. And of course, nitrogen, we do apply that annually, but that's very soluble and it can get down into the root zone very quickly. These other nutrients, phosphorus in particular, it's very difficult to move through the soil profile. So we need to have that incorporated before we plant. That's why we have to maybe build up a little bit of a bank, particularly for longer-lived crops like raspberries and particularly blueberries. So whenever these soil tests are done and recommendations come out, every soil is probably going to give you a slightly different response curve to the crop of interest. And we can't develop response curves for every soil type in your state, so we have to make some generalizations. And also, the truth of the matter is these response curves have not been developed for strawberries and raspberries and blueberries on all different soil types. Oftentimes, the agronomists have done these things for field crops, but not so much for fruit crops. So I guess the point here is that we're making educated guesses about how berry crops respond, often based on the response of some of these agronomic crops that we know a little bit more about. One approach that's often used is to find an agronomic crop that has a similar response to a fruit crop, but where there's a lot more data for, and use that as sort of a foundation for some of the soil testing recommendations. And a good example would be alfalfa, which tends to have the same sort of a response to nutrients in the soil and soil types as do strawberries and raspberries. Alfalfa is a perennial, strawberries and raspberries are perennial. They have about the same rooting depth. They have the same pH requirement for soil for optimal growth. So those are oftentimes used as a basis for a recommendation. Of course, people do nutrient response curves for some of these, and this is an example actually from strawberry, and this is looking at nitrogen. And these are looking at various rates of nitrogen fertilizer on plants, and this is in a liquid form, so it's not quite easily to convert to pounds per acre. But the point is, at low nutrient levels, you don't get much of a response. But as you move up, you get a higher response. But then the curve kind of flattens out. If we look at leaf area, we fertilize with more and more nitrogen. The leaves love it, and they really expand a lot and grow a lot. But if we look at yield, it's not nearly as responsive to the increases in nitrogen. So that says, okay, the optimal rate is probably somewhere around this 10N situation here. So we can do these response curves to some extent and sort of calibrate our soil tests by that. So it's not that they don't exist for berry crops, it's just that there's not a lot of those out there to calibrate on. Some recommendations are based on response curves that are dependent on the soil type. In other words, to get a good recommendation, you have to input the soil type. so that then the software knows what soil type that is and can make a recommendation based on that. A Morgan extractant is a good example of a type of soil test that is, you have to have the soil type, or at least know whether it's a sandy loam or a silt or a clay, because the response curve in the extractant is based on that soil type you have. Many labs are getting away from those tests that are dependent on soil type and looking at other tests that's less important. And the modified Morgan is one of those tests that is a little bit less dependent on knowing the type of soil that you have in there. And a lot of this is due to the amount of potassium that the different extractants pull out of the soil. But you have to sort of assume that the soil tests that you're using, the procedures and all, are good and accurate, and that the recommendations come out are on target. Now, a question was asked like a week or so ago about organic versus conventional recommendations. The recommendations that come out should be mostly the same, whether one's organic or conventional. It's not what's recommended that should be different. It's the amount and the type of product that's used that's different. So a soil test might recommend 50 pounds of potassium, but the source of that potassium is what would differ then between the organic and the conventional grower. There is some difference in amount based on release rate. Some organic nutrients are a little bit more slower to release their nutrients, so you may have to apply more of that because the release rate's slower. But in general, it's the total amount would be the same. So when you get your soil test, the most important adjustment that we usually make is to soil pH. If the soil pH isn't within the desired range, then the ability to take up nutrients will be compromised. So if you don't have the pH right, you can put on a lot of a certain nutrient and it might not even get into the plant because of the pH. And you've probably all seen this in soil classes back in college, but the amount of nutrient that's available to the plant is very dependent on soil pH. And the optimal range for most crops is a little bit less than 6 up to about 6.5. And of course, you all know blueberries are the exception. They're down around 4.5 for the optimum. But for most of the plants that we're dealing with the berries, and even vegetable crops, it's up around 6 to 6.5. Because of that range, most of the nutrients are reasonably available to the plant. Now, if we have to make an adjustment to the pH, we use sulfur to lower it and lime to raise it. Oftentimes in catalogs, and particularly for homeowners, you'll see aluminum sulfate recommended to lower the pH. And we do not recommend that because aluminum is toxic to plants at high rates. Now, granted, it provides a rapid change in pH because the sulfur is already oxidized when it's in aluminum sulfate form. So it could go right to work and lower that pH almost right away, whereas sulfur takes some time. But you don't want all that aluminum, so we don't recommend that for lowering pH. And changing the soil pH after planting is extremely slow and difficult. That's why it's so important to do it before planting. I have a couple of photos here of people who have planted and then realized that their pH was too low and tried to raise it with lime. And you can see it just makes a real mess in the field. It's really hard to incorporate and get that lime down into the root zone where it has an impact. So if you can mix it in the soil before you plant, it just makes a world of difference. Raspberry field, same way. How are you going to work that lime down into the soil well enough to really change the pH around the root zone to where those nutrients can be taken up better? Really hard to do that. So the amount of lime that you're going to apply to the soil to change the pH is dependent on two things. And one is the difference between the current pH and your target pH. So how big is the gap between where you want to be and where you currently are? And then the second thing is something called the cation exchange capacity, or the soil buffering capacity. This is the ability of the soil to hold on to the cations in the soil through these exchange sites. They're like negative charges on the soil particles. And it's the ratio of the cations that are bound to these negative charges to charges that only have hydrogen attached to them. It's the ratio of the cations to the hydrogen ions to determine your pH. This is often called base saturation, or the percent of the exchange sites occupied by the bases. So just think of it as like a column. The column has a lot of negative charges on it. Some of those charges then attach to positively charged cations, such as calcium, magnesium, and potassium. Those are the major ones. The remainder tend to be populated with hydrogen ions. And the more hydrogen ions you have, the lower your pH. Remember, pH is the negative log of the hydrogen ion content or something like that. Anyway, the higher the ratio of the hydrogen ions to cations, the more acid the soil. So the more of these exchange sites we have, and if we want to change it and knock some of those hydrogens off the exchange site, we have to add a lot more base. The negative charges on our soils arise from either clay particles or organic matter. Those are the sources of the negative charges. So the clay particles and the organic matter hold on to those positive charges that are out there. Soils that have a lot of exchange capacity, in other words, have a lot of negative charges, are more difficult to adjust the pH. So if you want to knock hydrogen ions off that exchange site, you have to use a lot of lime. But once you make that adjustment, soils that are high in cation exchange capacity tend to be more stable. They're a lot more resistant to change once you make the change. Soils that are low are easy to change, but they'll revert back to a low pH as those calcium and magnesium and potassium ions leach from the soil, and then the hydrogen takes over those exchange sites and the pH drops again. So to reiterate, it's easy to raise the pH of a sandy soil with low organic matter because sandy soils and low organic matter soils have low cation exchange capacities. Conversely, it's hard to lower the pH of a clay soil having a high organic matter content because both clay and organic matter contribute to these negative charges in the soil. So the amount of sulfur, this is just estimates, ballpark estimates, to lower the pH to a desired level of 4.5 depends, the two things we mentioned, it depends on the current pH, which is over here, and of course, the further we are from 4.5, which is our target pH, the more sulfur it will take to lower it. And the higher our cation exchange capacity, the more sulfur it will take to lower it. So in a sandy soil, not much. Loamy soil with more negative charges, higher cation exchange capacity, it takes more sulfur. And with clays, even more sulfur. So when we're up in this range here, with a high pH soil with heavy soil, it's really, really hard to lower that pH to 4.5, even with a lot of sulfur. It just takes a huge amount. So oftentimes soils up in here aren't really suitable for blueberry production. So soil tests attempt to estimate the amount of lime or sulfur required to bring the pH to within the desired range for a particular crop. And it's based on this cation exchange capacity. Soil tests do not measure cation exchange capacity directly. In other words, it doesn't go in and directly measure the amount of negative charges that are there and how many of those negative charges are populated by hydrogen ions. They use indirect methods to measure that. It's an inexact science. Different procedures are used in different labs, and the cation exchange capacity itself varies somewhat with pH and other factors. So, I guess what I'm trying to say here is that oftentimes the lime recommendations, we think of them as being really, really precise, but the truth of the matter is they're estimates. They try to put you in the ballpark, but they're not absolutely precise. The cation exchange capacity varies with various soils. Sands, of course, have low, and loams a little bit higher, and clays have higher, and you can see that reflected in the sulfur recommendations in the previous table. Humus, or organic matter, in its sort of broken down form, has very, very high cation exchange capacity. So that's why soils high in organic matter require a lot of sulfur or lime to make adjustments to them. And we don't want to forget that adding lime or sulfur to the soil does not change the pH instantly. It takes 6 months or longer usually. It's dependent on the size of the particle of the lime and the sulfur. Smaller particle sizes adjust more quickly. It depends on soil moisture. In dry soils, you get very slow oxidation and conversion. It takes a lot of time. You have to have adequate soil moisture to make sure it happens relatively quickly, and in temperature. In the wintertime, not much happens in the soil. That process of oxidation is a biological process. It requires soil bacteria to make that conversion. So when it's cold, there's not a lot of activity. Also, those bacteria have to have aeration. It's an aerobic process, and the soil has to be aerated. So all those things have to happen for that pH to change after we add our lime or sulfur. So now let's get into the crux of the matter here with that background on interpreting soil tests. We're going to look at soil pH, the macronutrients, the micronutrients, and I'm going to run through 7 or 8 examples, and we'll work through those and see how a soil test might interpret those and recommend those. This is just an example of a soil test. We'll look at some of its various parts. Of course, the reports that come back vary a lot from lab to lab, but let's just assume this is sort of a general recommendation. The first thing you want to look for, at least I do, is the pH. And we want to look and see whether that's within the ballpark range or not. And in this case here, it's for strawberries. The pH range that we use before we provide a lime recommendation is between like 6 and 6.8. So the pH is within that range, so we wouldn't recommend lime here. Sometimes you'll see a different pH number there. And in this case here, there's this thing called buffer pH in water. And people say, well, what is that? Does that mean my pH is too low because it's less than 6? Well, no. In this case here, this soil testing lab is using something called the buffer pH to estimate cation exchange capacity. And the difference between the buffer pH and the normal pH in water is an estimate of the cation exchange capacity. And there's complicated curves and correlations and so forth, but suffice it to say that the greater the difference between the pH and the buffer pH, the greater the cation exchange capacity. Therefore, the slower or harder it's going to be to change that pH if you had to change it. In this case here, we wouldn't have to change it. But in the situation where the buffer pH differed greatly from the pH in water, that would imply a high cation exchange capacity. So we don't worry about that for interpretation. We assume that the software takes that into account and spits out that lime or sulfur recommendation based on that difference, and we don't have to worry about it. But it's often shown there so you can see that it's there. And it gives you just some sense of, when you're comparing different soils, what that cation exchange capacity difference is if it doesn't print it out directly on the form. Some soil tests print it out directly. This one prints out that buffer pH to pH comparison. Another thing I like to look at is the organic matter content. I like to see organic matters, 2% or higher. This one's pretty good, 4.6%. It indicates that the cation exchange capacity is reasonably robust, and there's organic matter in the soil, and the plant roots tend to like that. So when the organic matter gets below 2%, then one of the things you would think about recommending to a grower would be some sort of a cover crop or manure application or something to boost that organic matter content. Soil tests generally report values for phosphorus, potassium, calcium, and magnesium. We talked about those curves before, those response curves. It's usually, they've been done for these 4 macronutrients and not for the micronutrients. So those are the ones you're going to get results from. Oftentimes these micronutrients will be printed out, but they're kind of hard to interpret. The techniques that are used to analyze nutrients are oftentimes something called an ICP. They send the extractant through that ICP, and it prints out not only the PK, calcium, magnesium, but it prints out everything else too. So it's not unusual to see a soil testing lab present values for zinc and manganese and iron, even aluminum. Most of the time, these aren't really very useful. Like, we don't use those for guidance in what to apply to the soil. Usually we use these when we want to try to diagnose a problem, like a toxicity or some sort of an imbalance. But they're typically not used to give recommendations on. Those are usually just pK, calcium, magnesium, and any sort of pH adjustment.
Marvin?
We also might see, yes?
Oh, I'm sorry. I didn't mean to interrupt.
No, go ahead.
Jeff had a question about pH when you're finished with what you're discussing.
Okay, let me read. So the question is, do we assume a natural reduction in pH over time and therefore apply lime to maintain a pH in a range of that over a 5-year period? So with normal rainfall conditions and all, we lose maybe 500 pounds of cations from the typical soil a year. So if you have a lot of calcium in your soil or potassium and so forth, we lose some of it, but it might not be enough to really knock the pH down even over a 5-year period. The time when we have to worry about that is on sandy soils where we can't really build up the reserve too much without pushing the pH too high. So if we push the pH to 6.5 and we have a sandy soil and we get a lot of rain year after year after year, yeah, it's true we could knock the pH down below a desirable level, and then we might have to go back in and reapply. But as I showed earlier, it's really difficult to do that in something like raspberries because they might live longer than 5 years. Strawberries, you just wait till the end of their normal life and reapply the lime, but on raspberries it's kind of hard to do that. So we want to try to make sure we get all that in there before we plant, so we don't have to go back in and try to add later on. I wanted to mention that the nitrates, some soil testing labs present a value for nitrate, but it really is so variable, almost from day to day, that it's not a very useful guideline for making recommendations. So this lab here doesn't even print it, although they would have it if you would ask for it. The other value we sometimes ask growers to run or make requests for is boron. Boron is one of those micronutrients that can have an impact on plant growth, and those values do mean something. So we will recommend that growers, particularly of strawberries and raspberries, ask for boron if it doesn't automatically come with the test. Sometimes it does, sometimes it doesn't. This is just a diagram of the different forms of nitrogen and the different cycles that are happening within nitrogen, and it's just so variable that anytime you do a nitrate or a nitrogen test of the soil, it's just a snapshot in time. It's so dynamic that those numbers really don't mean anything. So we don't pay much attention to them when we get them. Boron, on the other hand, I mentioned is an important nutrient. It's important for cell elongation. And if you don't have boron, your cells tend not to elongate very well. And so this is a root of a strawberry plant grown in a nutrient solution. It's complete, has everything in it. The next slide I'm going to show you is one where the boron was removed from the nutrient solution. And you can see how the roots are real stubby. And of course, a plant without boron has a difficult time taking up all the other nutrients that it needs. That's why we want to make sure we have adequate boron in the soil so you don't get a situation like this where the soil tests will show, well, you got adequate potassium and phosphorus and magnesium and calcium, but the plants are showing deficiencies in those values. Well, it might not be too low of potassium or too low of phosphorus. It might be that your boron is inadequate. That's why we want to look at that particular nutrient. Okay, back to the soil test. The other thing we want to look for is the type of test that we've run. In this case here, it was modified Morgan, but different labs run different tests. So when you get the values under there, they don't really mean a whole lot. If somebody sends me values of a soil test, I don't really have any basis for knowing what's low, medium, and high. You have to trust that the soil testing lab knows what low, medium, high is for your particular crop. So here we have recommendations on what low, medium, high is. And again, because different labs use different methods, different extractions, you got to trust that they know what they're doing. Now, and then you get your recommendations printed out down here based on the soil test levels above. I mentioned you have to have confidence in the recommendations provided by your lab. The best analysis in the world is useless without a good recommendation. Unfortunately, many analytical labs provide these general plant recommendations that are good for field crops, but they don't really fine-tune them to the needs of the specific crops that we might be interested in, like strawberries, like raspberries. And I can't tell you how many times I've seen soil tests come back for blueberries, and the recommendations look like they should have been for corn or something, because the lab is just spitting out a general recommendation for a generic crop on that soil without understanding the differences between these perennial crops and some of the crops that require a low pH. So just a word of warning on that. You've got to trust that the lab knows what they're doing.
Marvin, Jeff had a question on boron. Is it mobile in the soil?
Boron is very mobile in the soil. That's one of those nutrients that you can come in and apply any time of the year, and you can apply it right to the soil surface, even right over the leaves, and it'll wash into the soil and get taken up by the roots and into the plant. So that's one that's fairly easy to correct even after the plants are in the ground. It's not like some of the other nutrients that are very difficult to move into the root zone. So let's look at a few examples here and see what we can come up with. This is an example of a soil test, strawberries on a clay loam soil. And we're going to trust that the lab gives us the right values for what's low, medium, high, and they're reporting these out. Medium phosphorus, high potassium, high calcium, low magnesium, boron has more than 1 pound, and pH of 5.2, relatively high organic matter. So what do you think a soil test recommendation might provide for this particular example? Give you seconds to look at that. Maybe you might want to jot that down somewhere and see what you can come up with. So the first thing I would do, without knowing anything about the lab or the extractor or anything, is look at the pH. And I see, oh, it's 5.2. So it's less than the 6 or 6.2 to 6.5 that we would ideally want for strawberries. So I would know that some sort of a pH modification is in line here. I also would look here and see that phosphorus is a little bit low. We might want to raise that a little bit. I also know that a lime contains calcium, but it also contains magnesium. Now in this case, magnesium is low. So one option I would have in this case, and the recommendation engine should generate this, is when they're recommending a pH change, they might want to recommend high magnesium lime. So you can raise the pH and provide your magnesium in the same step. So here the recommendation would be dolomitic lime because it has high magnesium and phosphorus.
Marvin, Glenn had a question. Without N represented on the soil test, what is the basis for the N recommendation?
I'm glad you asked that question. The basis for the nitrogen recommendation is sort of a ballpark figure of what you would apply to a crop in that particular year. So for example, strawberries, a standard recommendation would be anywhere from 70 to 100 pounds of nitrogen per acre, just based on the age of the plant, based on the crop itself. We always have to add nitrogen every year, and that's sort of a standard. So it's not based on the soil test. That nitrogen recommendation is just sort of a standard practice that a grower would use to fertilize, to put you in the ballpark. And we're going to talk in a couple of weeks about using foliar analysis, and one of the most important uses of foliar analysis is to adjust those ballpark nitrogen recommendations to fit your particular crop situation. So the recommendation might say, well, an established strawberry field, 100 pounds of nitrogen, but we can use that foliar test to adjust that up or down based on what the values are in the leaves. We'll talk about that later. So the recommendation of nitrogen on a soil test for a perennial crop is just ballpark figure. This is what you typically do the first year of the planting.
Sonia is commenting that low magnesium can also be exacerbated by high potassium because they compete for uptake, correct?
There is some some thinking that can indeed happen. There's competition between these cations for exchange sites. So in soils that are really, really high in one of the cations and really, really low in the other, there's some sense that the high cations can suppress the uptake of the lower level of the cations in low supply. So yeah, those situations are pretty rare, but they can happen. And I'm actually going to give you an example of this in a few minutes.
Dan is wondering, on some of the high lime soils in central New York, where the pH values can be closer to 7, if an application of sulfur should be made to lower the pH for strawberries.
I would say, sure, certainly. Our target pH would be 6.2 to 6.5 for strawberries, and if you're up at 7 and you're in a pre-plant situation, I think sulfur would be an appropriate material to apply to lower that a little bit so that the nutrient uptake can be improved and you get less precipitation formed between some of these nutrients that happen at high pHs where they become unavailable to the plant. Okay. Let's look at a second example. This is blueberries on a sandy loam soil, and we have low calcium. We have pH that's just about right, high organic matter. So, what would we do in this situation here? Potassium's high, phosphorus is high, magnesium is high. It looks like we need calcium, but the pH is already kind of where we want it. We don't really want to raise the calcium too much more. What would we do here? So in this case, we might recommend calcium sulfate instead of lime to provide our calcium, because calcium sulfate doesn't change the pH very much when it's added. So it's a way of providing calcium without a big change in pH. So again, the soil test recommendation should know that, take that into account whenever it makes the recommendation. So I see Jeff. Okay, yeah. Go ahead.
Oh, no, I was just going to say Jeff had a question about sulfur.
Yeah,
the requirements for strawberries.
Yeah, sulfur is needed by alfalfa, but it's also needed for all other crops as well. It's an essential nutrient. It's part of amino acids, so all plants need sulfur. And in our part of the world, sulfur is rarely limiting. It comes down in rainfall, acid rain, and there's usually plenty of sulfur. It's rare that you have to add sulfur for the nutrient value to the plant. We might add sulfur for the pH adjustment, but usually not for the nutrient value. Again, there's sulfur around, it's in the organic matter. There's usually, we're not in situations here where we're usually very sulfur deficient, at least not in berry crops. Take a third example, raspberries, loamy soil. Here we're high and everything, the pH is right. Would the soil test recommend anything here? Well, it might, and this goes back to what Sonia asked about earlier. Sometimes, even though we have high values, if we're really super high in one nutrient, one cation in particular, it can depress the uptake of the other two. Particularly, calcium and magnesium can interact a little bit. Potassium, even though it's low here, that's actually a pretty high value for potassium, but calcium relative to magnesium is low. That ratio of calcium and magnesium is around, so higher than 10. Typical soils, it would be around 5 times as much calcium as magnesium. So in this case here, we actually might need to recommend a little bit of magnesium to bring that balance back up. So it wouldn't be unusual for a test like that to have a little bit of magnesium sulfate in the recommendation. Not so much to change the pH. This doesn't really change the pH, but it just adds a little bit of magnesium in the soil to bring that ratio into balance. Okay, example 4, blueberries, sandy loam soil. You can see that we're low in phosphorus, high in potassium, low in calcium. Magnesium is medium. pH is 5.9, and organic matter is pretty low here. So what would you recommend in this case? Or what do you think a soil test recommendation should say? One of the things we would want to do is try to raise that organic matter a little bit. Blueberries like organic matter. 1.3 is really quite low, so we want to do that. But the pH is 5.9. We have to lower that quite a bit. And one of the things we know that about blueberries is that they don't really require a whole lot of some of these cations. They're really good at sucking that out of the soil. So all we might need to recommend here is just to lower the pH, increase the organic matter, and those other nutrients will probably take care of themselves because we're measuring that at a high pH. When we lower the pH, for blueberries become much more efficient at taking these nutrients up. So I see a question here about how strong is the effect of added nitrogen on pH. It depends a little bit on your nitrogen source. If you use ammonium sulfate as a nitrogen source, which we often recommend on blueberries, it acidifies the soil. If we use urea, it also acidifies the soil a little bit because it's adding NH4 plus to the soil. But if we add nitrate to the soil, like calcium nitrate, it tends to either be neutral or raise the pH a little bit. So it depends a little bit on the source of nitrogen as the impact on pH. But ammonium sulfate is going to have the strongest negative effect on pH. Urea somewhere in the middle. Your nitrate forms of nitrogen, not much. But we wouldn't use nitrogen to actually try to affect the pH. We would use it to maybe keep it at a certain level. We wouldn't use, say, ammonium sulfate to lower the pH 1 unit. That would be way too much. And the question is, can blueberries use nitrate? They can use nitrate, but they don't like it. They're much more preferentially use the ammonium form of nitrogen and take that up. If you put blueberry roots in a solution that just has nitrate available to it, the roots will turn black and the plant will die. If you put blueberries in the field and you apply nitrate fertilizers to it, the bacteria in the soil under low pH conditions, which you should have if you're planting blueberries anyway, will convert that nitrate into ammonium and where it can be taken up more readily and used by the blueberry plant. So you can get away with it, but nitrate is more leachable, and also ammonium is a better source because it not only lowers the pH, but it's more of a slow release, more available over a longer period of time, and blueberries can take it up directly. So that's a preferable source for blueberries. Okay, just a couple more examples here. Blueberries, clay loam soil. We're high, medium in phosphorus, high potassium, high calcium, high magnesium, good boron. pH is pretty high, organic matter is pretty high. What would we recommend here? Wanted to grow blueberries. Well, if we go back to that chart we looked at before, and we saw a pH up around 6, and we see here that the cation exchange capacity is probably going to be pretty high because the organic matter is going to be pretty high. We're already high in calcium and magnesium, and we don't want a lot of calcium and magnesium in the soil. Blueberries don't like calcium in particular. The best recommendation here is probably just don't grow blueberries. The soil is just not appropriate for blueberries. The cation exchange capacity coupled with the pH is so great that it would take so much sulfur to lower that it probably wouldn't be worth it economically. And that high calcium is going to cause problems for years on end because of that. A couple more. Strawberries in a sandy soil, low phosphorus, low calcium, low pH, low boron, low organic matter. So this soil test is probably going to come back with several recommendations. Calcitic lime because we were low in calcium, some phosphorus, some boron, and some organic matter. And final example here, raspberries in a loamy soil or medium in most things. Fairly high in aluminum, good boron, high pH, organic matter. Now most of the time, if you saw a soil test like this, you might say, well, I've got medium values for most of these things. Organic matter is pretty good. pH is in the range I might want it. Probably don't have to do too much here. But in this case here, we might want to correct that with a small amount of P and K, but we might want to add a little bit higher rate of lime than was recommended in the soil test. And the reason for that is this aluminum value is pretty high. And that can cause problems later on. And the higher we raise the pH, the less available aluminum is. So you might want to push the pH to the very high end so that it makes the aluminum less available to the plant. And aluminum can be toxic. That's why we don't really want it. It can be toxic. So in this case here, the aluminum value is providing diagnostic information to the plant to understand maybe why those plants aren't growing very well, or what we might want to do to make a correction before we plant. This is a field of blueberries. This is a real situation, and the blueberry plants in this section here weren't growing so well. And the other thing is the grass in this section wasn't growing so well either. And yet down here at this end, the grass is growing pretty well. Now, we took soil tests from 3 different places from plantings that were 3 different ages. In one place, the plants looked pretty good. The sod was fine. pH was good. Aluminum phosphorus was okay. The other place, the plants looked really pathetic. The pH was a little bit higher than average. Aluminum was pretty high. Plants were pretty sick, and there was dead sod. And here, this is the interesting thing. The pH is really high. You'd think blueberries might not do so well there. And they were kind of fair. They weren't terrible. And the sod was living. These plants with a higher pH were a lot better off than the plants that were sick at even a lower pH. What was going on here? Well, it appears from the soil test that in this case here, aluminum was really high, and it could be becoming toxic to the blueberry plants. And as we raise the pH, even though the aluminum was up a little bit, the higher pH makes that aluminum unavailable to the plant, so the plants are able to survive a little bit longer and do better. So pH can react and relate to a lot of different things. And looking at these values of some of these micronutrients can give you clues sometimes as to how things are working out there in the soil. Last thing, the last concept I want to mention is that this nutrient uptake occurs through 3 different ways when we have nutrients in the soil. Direct contact with roots. So the root surface actually almost has direct contact with the soil, and the nutrient just kind of jumps right across that membrane. Mass flow, which is nutrients dissolved in water that get taken into the plant through the transpirational stream. Or diffusion. And diffusion is sort of short distance transport of these nutrients when they dissolve in water. The water actually doesn't move through the plant like mass flow, but the nutrients diffuse through an aqueous solution from a level of high concentration in the soil to a lower concentration inside the root. So there's 3 different mechanisms. And nutrients that are primarily taken up by contact or diffusion must be in close proximity to the root itself. The root on average only has about 1%, and at most 3% of the surface of that root is in direct contact with the soil. So nutrients have to be thoroughly mixed in order to be taken up prior to planting. This is with corn, but it's appropriate for all of our crops. This is the mode of uptake for the various nutrients based on this root interception, mass flow, and diffusion. And different nutrients have different modes basically being taken up into the plant. Nitrogen is mostly taken up by mass flow. In other words, nitrogen, nitrate, nitrogen, and ammonium nitrogen get dissolved in the aqueous solution. When a plant transpires, it moves into the root system, and the water leaves the leaf, leaving the nutrient inside the plant. That's why you can put nitrogen on the soil surface, and it gets down into the roots because it dissolves in the water, it's soluble, and it's taken up by mass flow. Someone also asked earlier about boron. Boron also is very highly soluble and taken up by mass flow. That's why we can apply boron to the soil surface. It gets dissolved, gets taken up by mass flow, no problem. We can apply it after the plants are established. But some of these other nutrients are mostly taken up by things like diffusion, like phosphorus. So it has to be thoroughly incorporated into the soil. It doesn't dissolve very quickly. It doesn't move through the soil very rapidly. So we want to have that mixed in with the soil before we plant. Same way with potassium. A lot of diffusion, not that much mass flow. So that's why magnesium, calcium, not much diffusion, some mass flow, some root interception, but certainly for P and K, we want to make sure we have that mixed in with the soil very well. Otherwise, the roots won't be able to take it up. These tables, of course, are going to be available to you to download, to print off, and it should be right here in this website in a few days when it gets posted. Now, once our plants are established and they're growing, we don't really rely on the soil test so much. We rely on the soil test to tell us what our pH is, but we don't pay much attention to the levels of P and K and magnesium and calcium after that. Once the plants are in the ground, we use a foliar analysis to make adjustments. And we use the pH in conjunction with a foliar analysis. And that's going to be the subject of an upcoming webinar. Next week, Eric Hansen is going to talk about these nutrient sources and some of the characteristics of these various nutrient sources that are out there that you can use in a pre-plant situation to both modify the pH and add phosphorus, potassium, magnesium, and so forth, and look at it from both the conventional perspective and the perspective of an organic grower. So that's what I have for the presentation, and there's a few minutes left. If anybody wants to type in some questions, we'll see if I can try to answer them.
Thanks, Marvin. I just want to remind everyone that if you haven't filled out the demographics questionnaire or the pretest, that they're available down below, and you can email them to me at mcm4@cornell.edu. If you missed either of the first two webinars, there's a link to them in the archive on the bottom left-hand side of your screen. And as Marvin indicated, Dr. Eric Hansen from Michigan State University will be our speaker next week.
So Jeff asks about organic sources of nutrients and their characteristics. That's what the entire webinar is going to be about next week. So we'll save that for Eric to talk about. An acceptable boron level, generally you want at least 1 pound, preferably 2 pounds. That's sort of the ballpark that we're looking at. And fortunately for boron, it's not so dependent on soil test. And it's also, most soil testing labs use a separate extractant for the boron. They use a hot water extractant. So they just put hot water in the soil, take it out, run it for boron. So the procedure for looking at boron is fairly standardized across labs. So we can pretty confidently say between 1 and 2 pounds is where you want to aim for. If it's less than 1 pound, you probably have to supplement. And that's going to be a lot more standard among labs than most of the other nutrient levels you're going to get. Jeff asks, is there a physiological reason for blueberries' aversion to calcium and nitrate? That's a really good question. So blueberries have evolved in these low pH soils, and nutrient levels are very low in these low pH soils, sandy soils, wet soils, and they've evolved some characteristics that enable them to survive under those conditions. And one of those adaptations is this association with endomycorrhizal fungi that colonize the roots of blueberries and then actually help the blueberry increase the surface area as they go out and grow through the soil. And the blueberry gives that endomycorrhizal fungus some carbohydrate, and in return, the fungus pulls in nitrogen and phosphorus and gives it to the blueberry plant. Most of the time, the nitrogen that's in the soil is in the ammonium form of nitrogen. And so some people think because of this endomycorrhizal association, the fungi prefer it, and the blueberries ended up just evolving a preference for that because that is the most available type of nitrogen in low pH soils. And actually, when a regular plant, a non-blueberry plant, takes up nitrate, interestingly enough, inside the plant, it converts it into the ammonium form and moves it around and then reconverts it into nitrate forms at times. Because if you look at amino acids and all that comprise proteins, they're in the ammonium form. So why, I guess maybe a better question is why other plants haven't evolved an adaptation to take up ammonium nitrogen, since energetically it's easier to do that than it is to take up nitrate and then reduce it back down to ammonium and move it around. And in terms of calcium, I'm not exactly sure why that is. I think it has to do with inside the plant interactions with iron, and somehow it maybe precipitates with the iron or inhibits it, but high calcium soils and blueberries just don't go well together, even if the pH is low. One of the things we'll recommend in a soil test, if the calcium level is above a certain amount, even if the pH is kind of close and you could lower it with sulfur, we might recommend look someplace else. This isn't a good soil for blueberries. You just know historically high calcium and blueberries don't go well together. Does soil moisture content at the time of sampling affect soil test results? Not really. Not if you're just looking at the nutrients, because what they'll do when they get the soil back in is they'll often dry it down anyway. And so whether you send them dry soil or they come to the lab and they dry it before they analyze it, doesn't make a huge difference. There's some debate whether there needs to be moisture correction to soils when they do the analysis, but that has more to do with some labs scoop soil and some labs weigh it. And the question is, do you get a different result when you scoop it or you weigh it, or you have it moist or it's dried? So it can make a slight difference, not so much qualitatively, but quantitatively. But labs have not come to consensus over whether it needs to be dried or not. Most labs do it dry. I think most labs do it dry. How do I feel about ammonium phosphates for blueberry? Ammonium phosphates are, if you're low in phosphorus and you need to fertilize the plant, that would be a good way of killing two birds with one stone. It provides an ammonium source of nitrogen, and it provides that phosphorus at the same time. And do blueberries have a higher iron content? I don't believe they have a higher iron content than other plants. That's not the case. But for some reason, iron availability seems to impact blueberries more than iron availability in other crops. Now, again, I'm not exactly sure why that is. Maybe somebody out there knows the answer to that.
Thank you very much, Marvin. I really appreciate you speaking to us today. Again, next week our topic is soil and nutrient management. And we'll be looking at correction of soil problems, application technology, conventional nutrient sources, organic nutrient sources, Lyman and pH reduction with Eric Hansen from MSU. A couple more questions, Marvin.
So the availability of calcium sulfate in New York, it's gypsum. I know that there are, I'm not specifically in New York, but drywall for housing is gypsum. And there are some places that I know are crushing that to extract the material from within the drywall and then selling that for agricultural uses, and that's a great source of gypsum. And the high iron content is related to the low pH, like with cranberry. Yeah, it is. I mean, if you look at those charts where we looked at the availability of nutrients with pH, you can see that at the lower end, iron availability goes way up. And for some reason, calcium inside the plant, not outside the plant in the soil, but inside the plant, has some impact on that availability of calcium. It could have to do with the fact that active iron in the plant is in the ferric form, and maybe with too much calcium in the plant, it converts more to the, it's in the ferrous form. It converts more to the ferric form. So it goes from Fe+2, which is active, biologically active, to Fe+3, which is inactive, and calcium may cause the iron to go from one form to the other, in which case a leaf analysis will still show you have iron in the plant because it doesn't distinguish between the two types. It's just that the type in high calcium soils is more in the ferric inactive form. So that's my hypothesis. Again, maybe somebody else knows.
All right. If there aren't any other questions, then I think we'll close. Thank you so much, Marvin, for your insights.
You're welcome.
Thank you, everyone, and we hope you'll join us all again next week.